Hi everyone. So I wanted to show you how to do some data analysis on your raw data that you collected in the lab yesterday. So first things first, I have the data that I collected from today. So I named that Kabomba sample. This is data that was collected using the Kabomba plant. So this is how it looks on my screen. And there's a whole lot of information here. Don't worry about it. Don't be too intimidated by it. What you're going to do is you're going to control or right click, select all. You could also do Control A to do that. And then you're going to copy all that information or Control C. Then what you're going to do is you're going to open up a brand new Google Excel or Google spreadsheet or Microsoft Excel form. You're going to click, I would recommend clicking on cell B2, and then you want to paste all that stuff you just got. And boom, all of it's here in the spreadsheet. There's a couple things I want us to fix while we're here. So first off, we should put some parentheses there. Second off, I don't like how it has the 2 and it won't let me do a subscript, so then I change this to say carbon dioxide concentration. Also, the ppm should be in parentheses. One neat trick you can do here is you can double click this right here and it will snap to whatever size you need it to be. So I'll do that for this one as well. Then what I'm going to do is I can type here and I'm going to say Kabomba data. All right, then I can take this, come over here, click this button, which merges those two. Then I'm going to come here, center align it. In fact, I'm going to click both of these. If I just click here, it's just going to do those two cells. But I want to click both of these, the whole entire columns, and I want them to be center aligned. I also want this to be easy for me to see, so I'm going to make those bold. Well, now I have to resize this a little bit. There we go. This got a little bit bigger than it needs to be. There we go. So, a couple things I would do with my data here. Some of these are just some formatting issues. Some of them are other things that I really think are important when you're doing this. So, first things first, what I want to put here is change in carbon dioxide concentration from beginning. Okay. Oops. Of course, we measured that in ppm. Make that bigger, bold, and then I can make this go across all of that. And so here's what we're going to do next. So I want this cell to tell me what's the difference between whatever this next number is and that. So for example, I want to know what's the difference between 312 to 313. Now you're probably saying, Mr. Boyd, that's stupid, that's easy, that's obvious. But here's where the power of the spreadsheet programs comes in. So what we're going to do is we're going to say that this cell right here is equal to this cell minus this cell. Now here's the difference. This second time where we see C3, I want that to always reference cell C3. So on your keyboard, you can hit F4. Or you can just type in $C, $3. Then I'll hit enter. These aren't center aligned. Now we go, center aligned. Now I'm going to, see this little square in the bottom? I'm going to grab that and drag it down. Now it says 1 because what this is saying is that cell C4 minus that C3 will tell me the difference. Now here's where that becomes powerful, is I can do that and I can drag this all the way down to the very bottom, this group ran their experiment for just over 10 minutes. And look, it does all that work for me, which is pretty cool. Right? Lots of data here. One thing that might be helpful just to scroll through the data faster is to zoom out so you can run through everything a lot quicker. Another trick I want to show you, which can be really helpful, is if you notice this darker gray bar right here, if you put your mouse over it, you get a hand. I want to grab that and I want to bring it down 1, 2. So it's right there, and this thick gray bar goes here. Now, what's going to be really cool is when I scroll, that'll stay there. So I can look at all the different numbers then. All right. So this is pretty cool. This is really neat data. Pretty neat stuff. One thing I want to point out is if we ran our experiment for 10 minutes, that would be 60 seconds times 10 minutes. So that means 600 seconds. That's when we hit the 10-minute mark. Okay. 10 minutes. So just like an FYI. So that means that we don't necessarily need to include this extra data. We can, but we don't need to. Okay? That's your choice. What I would do then is I would select my data and I would look at this. And so this tells me that between the beginning of my experiment and the end of my experiment, that 120 parts per million of carbon dioxide were taken in by the plant and were taken out of the air that was in the container. This supports my idea that photosynthesis was occurring. So that's really cool. But I asked you guys to do some line graphs and some different things there. So what you can do is you can select all this data again. Okay. Then you could go to Insert, Chart. And it's giving me some stuff already. It automatically put it in a line chart. It automatically put, it kind of messed with some of my things here. So let's try and click on this and see. Oh, no, that's not what I wanted. All right, so that's exactly what I wanted. Use column B as labels. So it said time. And remember, I went to 600 minutes. So I know right away that 0 to 600, that's the time that I'm looking at. And then this was the concentration. So this is going to tell me all that stuff. And if I put my mouse over different parts, it tells me all that information. So what I would do from here is go to Customize. And then here you can give your chart a title. That's a pretty okay title. I think something better would be carbon dioxide concentration, and that's our dependent variable, and our dependent variable changes because of our independent variable. So carbon dioxide concentration as a function of time, because that reinforces then for my reader that carbon dioxide concentration changes because of the amount of time I let go by. Now you can also change the horizontal axis here. So I want this to be time in seconds. I want my vertical axis to be concentration of carbon dioxide measured in ppm. All right, and so you can see that coming there. You can also, there's no reason to really label the series. If you'd like to change the color, that's fine. Another thing we can add is a legend. Again, you don't really need a legend here since we just have this one big set of data for now. You can change the vertical and horizontal axis. Not a whole lot you would really want to do there. You can do minor grid lines if you'd like. They're kind of not that important though for this. So I would just leave that alone. I would leave this graph as it is. It looks fantastic, if I do say so myself. And if you wanted to, you could then move this to its own sheet, and then you have this whole page. You can notice down here it says chart 1. This is that whole thing. One thing you could also do if you wanted was, I don't know why that looks kind of weird here. Let me come back and see if that changes. No, I don't know why. But what we can try and check out here is see if this will let me do a best line of fit, just to see what kind of information it gives me. It could be a good idea to include this information there. And so that way it tells me, hey, this data was really good. It tells me that was almost 1. Remember, an r squared of 1 is perfect. An r squared of 0 means it has that the two are not related whatsoever. This is pretty good data. So if this was your group, great job. Coming back to this data that I have here, what I'd also recommend doing is on the whole class data, then you're going to add the basic information that you put together here. So the basic information for this group would be what condition, so that's the color of light, what was the change in concentration, and for this group, we noticed when we went all the way down to the bottom, again, it's a little easier if you do it this way, grab this, scroll to the bottom, and then zoom back in, was negative 120. If we kept the experiment going, and again, this group could perfectly do that. There's no problem with that. They're just incorporating more data. Then it went to negative 138. So then it went down by 120 ppm or 138, depending on how far they ran with the experiment. But this is the kind of information your group, the whole class will need from each other, because then we can take this information and really plot it out and see how all the data compares to each other. All right, on Friday I will be here to talk about how to combine all the class data. So for now, just set up this information for your group, save it, share it with all your group members, and we'll pick up from there.